So it looks like we have time for the complex biologic considerations. I find this, for me, the most interesting part of the talk. So, all the different biologics out there have different considerations for immunogenicity. Monoclonal antibodies are the most common biologic that's out there. Obviously a fully humanized antibody, which is the most common type of antibody that's being produced now, definitely lowers the risk of inducing ADA. But the mechanism of action of the therapeutic also plays into how you're thinking about, a monoclonal antibody. Is it immune suppressing monoclonal antibody, or is it immune stimulating, modulating antibody? So, and how is that going to affect your incidence of ADA? From the literature, immune checkpoint inhibitors, which are, stimulating the immune system, they tend to have a low incidence of ADA. So, from what I've seen, just because you're stimulating the immune system, you're not necessarily getting a higher incidence. And I think it comes down to more about how immunogenic that monoclonal antibody, the CDR region, is for patients. And definitely, B cell depleting definitely have low incidence of ADA. For ADCs, you have to think about, the agency wants you to be looking at all components of the ADC. And so you might have to have multiple assays for evaluating immunogenicity for an ADC. You want to think about the linker. From the literature I've looked at, the hapten-like structures the linker structures don't appear to be increasing the immune response. But something else you have to think about with ADCs is, the critical reagent of the drug itself can lead to problems when you're developing the assay. So you have increased hydrophobicity, you could have aggregation and stability issues, and so you have a hard time developing an assay to this molecule. So it's something to think about. So endogenous proteins, these are whenever I'm working on either fusion protein like FC fusion protein or endogenous proteins, they're the most risk. Historically there have been issues with with EPO is a famous one, where you can have devastating consequences to either healthy volunteers or patients. The risk is, it doesn't happen very often, but when it does, it can be quite devastating. And so with this, you're going to have a more thorough and thoughtful immunogenicity strategy, evaluation strategy. You need to think about the patient population. Is the protein absent? Is it truncated, or is a new epitope being generated that may be immunogenic to this patient population? You want to do frequent monitoring, and then even domain mapping of the ADA response, which part of the molecule is causing immunogenicity. Fusion proteins is basically the same thing. You just have the IgG region, which you're not going to typically have an immune response to the IgG region. It'd be to the linker or to the fusion protein itself. And you want to make sure your ADA assay and the anti-idiotypes that you developed, for your positive control in the ADA assay are specific to the active region of the protein. Peptides are, tend to be less immunogenic because they're smaller. Anything above 40 amino acids filed under a BLA. Anything under is filed under an NDA. You got to think about the size, the structure, the modifications. If it's conjugated to PEG, you're going to need an ADA assay against the PEG itself. And you got to think about, aggregation, same with the ADA or ADC. How does that critical reagent is going to be onerous to develop an ADA assay. So when I think about gene therapies, I think I'm going to be developing a lot of different ADA assays. You need to have, a 3-tiered plus NAB against all the different, the vector, the cargo, and the resulting therapeutic. So, there's going to be a lot of ADA assays. You want to start this early and resource it appropriately. And so, and also something to think about is patients with genetic disorders may not have developed tolerance against the transgene, and so the gene itself, it's kind of like a fusion protein or a protein therapeutic, in that you may develop an immune response to it because it's something the body's never seen before. With CAR T therapies, ADA has been linked to a decrease in efficacy in the past. So autologous CAR Ts are going to be patient-derived, and they're going to be less immunogenic than, say, allogenic donor-derived CAR Ts. The risks are inflammatory reactions and tox, and you usually see the immunogenicity against the transfected receptor, and it has been linked, as I mentioned, as a high risk factor for not having efficacy in the patients. I have seen both plate and cell-based assays being developed. So not like a NAB assay, but you would actually, your ADA assay would be flow-based, and it would be a cell-based assay for ADA. You're going to have, you may not meet guidance with this in terms of, your percent CVs. So you may have to have a larger window for acceptance criteria. You have to develop a cell line. The benefits of having a cell-based assay is that you have the whole tertiary structure being expressed as you would expect, versus a plate-based assay. You could have aggregate formation. The extracellular receptor that you developed as your target can be insoluble. And you have to label your reagents here, and that could mask the epitopes. So cell-based, there's benefits to both the cell-based and a plate-based assay, and you have to figure out what's going to be best for your program. Oligonucleotides, they're still required to have an ADA assay and immunogenicity assessment. They tend not to be very immunogenic, and a lot of times they're delivered to immune-restricted sites, say for like Macogen, but they're still required. You're still required to have some kind of immunogenicity assessment for oligos.